Inflammation is the body's fundamental defensive response to injury, infection, and tissue damage. When you twist your ankle and it swells, when a bee sting turns red and hot, when you get a fever with a respiratory infection, in all these cases, what you are experiencing is acute inflammation, a carefully orchestrated biological program designed to contain damage, destroy pathogens, and initiate repair. The cardinal signs of acute inflammation, redness, heat, swelling, pain, and loss of function, were described by ancient physicians thousands of years ago. What they could not see is the cellular and molecular machinery that produces these signs. Today, we understand inflammation not only as a protective mechanism but as a process whose dysregulation, becoming chronic, low-grade, and persistent, underlies some of the most common and devastating diseases in modern medicine. Acute inflammation begins when pattern recognition receptors on immune cells, primarily toll-like receptors, detect molecular patterns associated with pathogens or cellular damage. These receptors activate a transcription factor called NF-kappa B inside the cell, which switches on the genes encoding inflammatory cytokines. Within minutes, the injured or infected tissue is flooded with signaling molecules. Interleukin-1 and interleukin-6 amplify the inflammatory response and induce fever by acting on the hypothalamus. Tumor necrosis factor alpha coordinates cellular responses and increases vascular permeability. Histamine released from mast cells causes blood vessels to dilate and become leaky. This vascular permeability is what produces swelling, plasma proteins, and fluid escape from the bloodstream into the tissue. Neutrophils and then macrophages migrate through the vessel walls into the tissue, guided by chemical gradients, to engulf and destroy the source of the problem. Resolution of acute inflammation is as active and tightly regulated as its initiation. As pathogens are cleared and cellular debris is removed, anti-inflammatory signals take over. Lipid mediators called resolvins and protectins actively promote tissue repair and signal the immune response to stand down. Neutrophils undergo apoptosis and are cleared by macrophages, which transition from a pro-inflammatory to a tissue repair phenotype. When this resolution program works correctly, the tissue returns to its normal state with minimal permanent damage. When it fails, when the inciting stimulus persists, or when the resolution machinery is faulty, acute inflammation transitions into chronic inflammation, a lower level, persistent activation of the inflammatory program that smolders in tissues for months or years. This chronic state is fundamentally different from acute inflammation in its biology and its consequences. Chronic low-grade inflammation is now recognized as a central mechanism in a remarkable range of diseases that on the surface seem unrelated. Cardiovascular disease. Inflammation in the arterial wall drives atherosclerotic plaque formation and the rupture events that cause heart attacks. Type 2 diabetes. Chronic inflammation in adipose tissue and liver impairs insulin signaling and contributes to insulin resistance. Alzheimer's disease. Sustained activation of microglia, the brain's inflammatory cells, produces neurotoxic cytokines that accelerate neurodegeneration. Cancer. Inflammatory microenvironments promote tumor growth, angiogenesis, and immune evasion. Depression. Elevated cytokines impair serotonin synthesis and reward circuitry as described earlier. C-reactive protein, or CRP, a protein produced by the liver in response to inflammatory cytokines, is used as a blood biomarker of systemic inflammation and is associated with cardiovascular risk independent of other traditional risk factors. Anti-inflammatory treatments span a wide mechanistic range. NSAIDs, nonsteroidal anti-inflammatory drugs like ibuprofen and naproxen, work by inhibiting cyclooxygenase enzymes, COX-1 and COX-2, which produce prostaglandins, lipid signaling molecules that sensitize pain receptors, cause fever, and promote vascular permeability. By reducing prostaglandin production, NSAIDs reduce pain, fever, and swelling without fully suppressing the immune response. Corticosteroids, like prednisone, act more broadly, suppressing NF-kappa B activity and reducing production of many inflammatory cytokines simultaneously. Their broad immunosuppressive effect makes them powerful but also prone to side effects when used chronically. Biologics, a newer class of drugs including TNF-alpha inhibitors like adalimumab and IL-17 inhibitors, selectively block specific cytokines, allowing targeted suppression of inflammatory pathways while leaving others intact. They have transformed the treatment of rheumatoid arthritis, psoriasis, and inflammatory bowel disease. The relationship between lifestyle and systemic inflammation is increasingly well documented. Chronic sleep deprivation raises CRP and interleukin-6 levels. Insufficient sleep is pro-inflammatory. Regular aerobic exercise, in contrast, reduces circulating inflammatory markers and induces the release of anti-inflammatory myokines from muscle tissue. Dietary omega-3 fatty acids, found in fatty fish and some plant sources, are precursors to anti-inflammatory prostaglandins and resolvins. Obesity, particularly visceral fat accumulation, is one of the most potent drivers of chronic systemic inflammation. Adipose tissue actively secretes pro-inflammatory cytokines called adipokines. Understanding inflammation as both a protective mechanism and a disease driver connects disparate areas of medicine into a coherent biological framework. The same molecular machinery that saves your life when you have an infection can, when chronically activated, undermine nearly every organ system in the body.